Allow me to introduce the Kids First Data Resource Center, which is led by Dr. Adam Resnick from the Children's Hospital of Philadelphia in collaboration with 5 other sites. Dr. Resnick is the co-director of Data-Driven Discovery and Biomedicine at CHOP, and I want to emphasize that the Data Resource Portal, which you will hear a lot about today, was launched just 1 year ago this month. So we are very excited to hear about its progress to date.
So thanks again, everyone, for joining. So as Valerie mentioned, we're essentially 1 year old today, or at least this month. And what we thought we'd do today in the context of this webinar is really introduce some additional features that we've been deploying within the Data Resource Center's architecture. But more specifically, I think as we have begun processing and deploying and releasing data sets, wanted to go through some of the features and workflows for team-based discovery that we can empower within the Resource Center. In alignment with the Kids First programmatic vision, really our goal is to drive collaborative research through the Data Resource Center architecture that really harnessed a data sharing capacity. So here you can see that the DRC platform status is fairly robust at this point in time from a data perspective, and will continue to grow over the coming years. But really trying to harness these data into an operational framework that supports research is our true mandate. And so today we'll really go through a use case scenario where diverse expertise and resources are being engaged in the context of an emerging project. Really, with each of us that is presenting today playing the role of individuals in the context of a research project, trying to leverage the resources within the DRC on behalf of hypothesis-driven research. And you'll see today as we move through the platform elements of really trying to harness FAIR-based principles of findability, accessibility, interoperability, and reusability, but recognizing that operationalizing big data into hypothesis-driven research requires very diverse expertise. We're trying to implement different features, portals, and environments that allow the discrete user base of our community to engage the platform. So in some respects, because of time limitation, it'll be a bit like a morning show, cooking show, where some of the things are already pre-cooked and come out of the oven as if they've already been baked for a long period of time, but should really be authentically representative of what one version of a user experience can be. And then we'll end with this slide in the context of the resources. But I'll turn it over now to Vincent, who will really, from St. Justine, who will demonstrate some of the portal functionalities and begin the demonstration with the building of a cohort.
Thank you, Adam. So I'm going to show you today the new features we have implemented since the last webinar a few months ago. So first of all, you can notice that for those who have seen the portal before, so we added the ability to log in using your researcher ORCID account. So this was requested by some researchers who were not allowed by their institute to use Google to log in, to sign in. So we had to add this feature, so I can do that. So I'm going to log in now using my ORCID account. Oh, okay, so I'm done. And then, so the portal as before, 3 sections: a dashboard, an exploration data, which is the Chord Builder, and a file repository. In the next few months, we're gonna add more features, two new sections, one for searching members and connect to them, and also a section on the variant database that we are starting to build for Kids First. So let me show you the Chord Builder. So this is the same Chord Builder as you saw before. You will notice that we just removed the beta version banner at the top of the page. Over the last few months, we integrated user feedbacks and we fixed some bugs. We added new features, and I think now we can say that this tool is complete and stable enough to be called version 1. So the chord builder has the same three sections. So you have a section for a filter section when you can select search criteria. You have a query section in the middle, and then at the bottom you have two panels, two views of the results. So a summary view with some visualization, and a table view which show the list of participants for the current query. So the new thing now is that those charts are clickable now. So for example, you can, if you're interested to the first study here, you can click on it, and then it adds a term in the query section, and everything gets refreshed accordingly. So now I'm looking at the data for the study congenital earth defect, the kids first one. And you can see at the right that you have 2,133 participants in that study. Now I can add the new criteria using the filter section. For example, if I'm interested to do a family analysis, I can look for search for only trios in that study. So I can do a search family here. And then I find the family composition field, click on it, and then I can select trios. So you see that for each fields displayed in this bar, you have the count on your right. This is the count on the number of participants for this category. So we have 888 participants in a trio in that study. So I can select the field, the value, and add it to my query. I can duplicate queries. So let's say I want to duplicate the current one. And then I get a second queries. And this is one of the nice features of this chord builder is that you can add more and more queries to elaborate complex ones. So it's kind of a process that you can implement in your way to search the kids first cohort. So let's add a new criteria, for example, here. So I can add a diagnosis if I want to be more specific. Let's say diagnosis. I can search for diagnosis Mondo. So we have ontologies for diagnosis and phenotypes. The different ontologies you can see for diagnosis of the Mondo one, the NCIT, and we kept the source text as well. So let's say I select Mondo, and I want, let's say I would select the hypoplastic left heart syndrome here. So 54 participants satisfied that this criteria. So I can apply. And you can see I have 54 on the right side. I can see I have 54 participants in that. I can always go back to the first query and then re-execute it automatically, and the result at the bottom gets refreshed again. Okay. A new feature we have is it's the ability to upload IDs. For example, if you know the participants you're looking for, or if you know family IDs, you can just upload it there. So for example, let's just start a new query here, and I will upload some IDs that I have in mind. So these IDs are 3 families IDs. And then, so the tool actually found all the participants belonging to these families. So I can, if I type an ID that doesn't exist, for example, this thing, I will have an unmatched, I will have a one line in the unmatched tab here saying that, well, this ID doesn't match. You can upload CSV, list a long files of ID if you want. So let's execute this query, view result. And then you can see, for example, in the table view that you have, 9 participants in these 3 families that I have just uploaded. So that's a good tool. So let me start a new query. So as you know, the kids first harmonize and integrates cancer and birth defect cohorts into a single platform. So which enables a lot of interesting research questions actually that we can answer with this. So for example, let's say my use case is to find all the cancer patients that have shared structural birth defect phenotypes with non-cancer cohort. So I want a mix of birth defect and cancer within the same participants. Yes. So let's do that. So first of all, I will find all the birth defect participant or participant from birth defect cohorts. So I can do in two ways. Either I select each study that is about a birth defect individually, or I can simply type here birth. Sorry. And then I can see that the system find a field called diagnosis category, and one of this value is structural birth defect. So let's select this. And then I can see now that I have 2,293 participants in birth defect cohorts. Now I can duplicate this query. And I want only those, among those one, I want only those who have a phenotypes, an observed phenotype reported. So I can go here and go to the observed phenotype, the HPO, the ontology HPO. Open all of them, and you can see I have 345 such a phenotype. So I can select all of them and apply the criteria. So now I'm down to almost more than 1,900 participants who belongs to a birth defect cohort, and for which we have whom we have observed phenotypes. Now the question is that, okay, are these observed phenotypes shared with any cancer patient? So I can duplicate the query again, and then here, instead of selecting structural birth defect, I will select cancer. And by doing this, what I find is all the cancer patients with observed phenotype that are shared with a patient from a birth defect cohort. And then I got 63 participants there. So at any point of time, you can actually save your result. And then you can save your cohort using this button and add give a title, a description, and you can do it. I won't do it because I already done it before, but you can do that and refer, and when going back to it afterward. Let me do another last query. So let's say I want to be more specific. I want to add some diagnosis. So I will open the, sorry. Yeah, that's actually a new thing I wanted to show. So we do have now histological diagnosis. So if I want to be more specific, I can search for histological diagnosis here. And you can see we have 3 histological diagnoses, Mondo and CIT, and the source text as well. So I can select source text. And then let's say I select the first one, so the low-grade glioma. And then I'm down to 13 participants with cancer, patient with an histological diagnosis called low-grade glioma, and with observed phenotype shared by some birth defect participation as well. So a new thing we have now also is a participant page. So you have a link on each participant ID, so you can click on these links and you open a specific page for the given participant. So you have a summary, you have 2 tabs, summary and clinical. So the summary contains a summary, who is this patient, a link to PET bioportal for cancer patient, and Adam will show it later. And then you have the list of biospecimens for this patient. Yeah. So, and you have 2 kinds of biospecimen, those with an histological diagnosis associated with, and those without any diagnosis. So, and then you can go there like this, and again you can have more details about the specific specimen, biospecimen. We have also at the bottom all the data available for this participant. So you see, for example, we have two Align reads files for whole genome sequencing. We have other data type, gene fusion, so forth. And when you click on those numbers, you will be directed to the file repository and you will see those files in the list. Another interesting thing is the clinical tab. This will give you a summary of all the clinical information we have for this participant, on this participant. So we have the diagnosis, which is a cancer patient, actually. And then you have the phenotypes for this patient. And then you can see here, interestingly, that this cancer patient has actually 3 congenital birth defect, HPO, actually, source text. And then if you want to see, for example, the second one is the congenital anomaly of intestinal fixation. And then you might want to know, okay, is there any other patient with phenotypes? And among them, is there any cancer patient? So you just click on the link here, and this will open the Chord Builder with this query term. So HPO equal is any of this intestinal malformation. And then you can see that you have 14 other participants with the same phenotypes. And among them, you have some cancer patient. You have 1, 2, 3, I think 4 cancer patient, and the others are structural birth. Comes from a structural birth defect cohort. So it's very nice integration of both cancer and structural birth defect data, and this is quite unique to Kids First. So now I'm gonna share if I go to my dashboard, actually no, I can reopen the cohort I had made before, so which is called cancer patient with shared phenotype with BD. So I can reopen the cohort. And this is the cohort I built with 62 participants at the end. And I can share it, and I will share it with Adam, actually. So Adam can take over, and it will show you some integration with the CBio portal. So if I do like this, I can copy a short URL and Slack it to him. Is that Adam? Yeah. And Adam can take that link and open it, and you will see my saved cohort. Yeah.
Perfect.
Okay, so I will.
Thanks so much, Vincent. So Valerie can switch to me. So I have the cohort that Vincent shared with me. And you can think of this in the context of someone, exploring like Vincent did, coming to a cohort that they discovered. Vincent may or may not be a cancer expert, and more specifically, he may or may not be a low-grade glioma expert. And you can see that here, the most frequent diagnosis on the right-hand side within this cohort of 63 is low-grade glioma. So this is where someone who's created a cohort can share potentially that cohort with an expert and say, what do you think of this, and what can you make of it? So someone like me may get that cohort and say, oh, that's interesting that there are low-grade gliomas that are part of this. Let's explore the low-grade gliomas here and what is available. So I would go ahead and click, as Vincent suggested, and add and see the 13 participants that are in this low-grade glioma cohort, and I'll click and see what is available for these 13 participants. So here you can see that these are 13 participants that have a variety of different features as well as files associated with them. So I can click between the two tabs and assess that they have whole genome sequencing and RNA-seq data. And I can also see that that these 13 participants are only a portion of all the low-grade glioma samples that are actually available in Kids First, prompting an obvious question of saying, what's different between the 13 low-grade glioma patients that have structural birth defects than the other 200 and odd subjects that do not? And in doing so, one can get there in a couple of different ways. As Vincent mentioned, you can explore specific participants individually and and look at them one at a time and view their process data in the context of PHC Bioportal. And that's what I'll do at this particular moment. So I just opened the very first low-grade glioma patient, and I'm going to click on the PHC Bio link. And that'll take me to the patient-specific view within PHC Bio, which allows me to at least view the mutational context associated with that particular patient. Now, if you're a low-grade glioma biologist, the first thing you would have expected is some already pre-characterized mutations in low-grade gliomas, and a biologist like myself might say, actually, I've expected mutations of X, Y, or Z, and I do or don't see them in this particular patient. That actually turns out to be true for this patient. Most low-grade glioma, for example, are driven by a gene fusion event with BRAF, and that is not annotated here. So I can further explore the rest of the low-grade glioma patients by just clicking on the entire cohort they belong to. This is an identical representation within PHC Bio of the cohort and its files within the file browser environment. And here you can see that there are 259 low-grade astrocytoma overall, and we already know that 13 of them actually have birth defects characterized. In PHC Bio, you can also begin making some comparisons and some analysis. So I'll just show some briefly. Here you can see if I click on low-grade gliomas as a whole, the most often mutated gene is BRAF. And we can actually assess whether or not the 13 patients that we highlighted are actually also fall into that category. So here I have actually a list of those 13 patients that I can export from Kids First. And I'm going to go to PHC Bioportal and put that list in to review this cohort. Before I do that, though, I'll actually create a group. You can see I've created some groups here before of all of the cohorts that are here, and I'll actually just change the name. You can see that I've practiced this particular one before. So this will be all. But then I can also, as I mentioned, create a custom group of just the patients that had a structural birth defect and look at those, right? And you can see that some of them have BRAF and some of them do not, but I can also click on individual ones and assess them. But again, I can also create another group of these patients to compare. So I'll go ahead and create a group here. Just 13 and create that. Once you can create groups, you can actually begin comparing them from one to another and do some initial visualization of those cohorts to just see if there's something of interest that you might want to define. So as expected, 16 samples from 13 subjects are part of a larger cohort. But once again, you can create groups that are more specific to those that overlap or don't. So you can say no overlap. And then you can create comparisons between these groups and analyze, for example, survival or any other features of interest for them. Now, the part that I think is really interesting as a biologist is that once you start looking at these cohorts, the interesting thing that you come up with is that the 13 subjects that you identified actually are not similar to other low-grade gliomas, and none of them, for example, contain the expected gene fusion event that drives most low-grade gliomas. So if you wanted to understand, well, what can you potentially target in this cohort if they don't have a canonical mutation, you would have to go back to the original data set and then perform additional analysis. The PHC Bioportal environment provides you the opportunity to essentially play with process data, but if you have a hypothesis, you may want to go back to Kids First environment and create additional analysis to move forward from that context. So again, what I would do is create an analysis. I would choose these 13 samples again, just like I did before. But this time, I would admit to myself, I'm not an expert at processing BAM files or CRAM files, and I would rely on additional partners to do that. So I would go ahead and select the type of files that I think are interesting and create a cohort that I can then push into Chavatica and share with partners who might analyze them. So I'm going to push it into a pre-composed environment because I already in my mind I have some comparisons that I would like to do in order to try and identify new targets in this new sub-cohort of patients that does not have a canonical drug target. So I'm going to copy these files to this GTEx comparison project and then Slack my partner Allison and say, hey, can you help me analyze these new files? But instead of looking for mutation, I'd really be interested in looking at the expression levels of these subjects and comparing them to normal expression levels derived from GTEx.
Great, thanks, Adam. So I will be playing the role of the technical bioinformatician, which I must admit is a role I haven't played for a few years, but I think, you know, towards Adam's point, you know, these handoffs between these different teams, you know, I think we saw Vincent use Slack and other things. We know that there's these kind of handoffs in these platforms, and you aren't just going to do everything from one place. So we really think about those different kind of workflows. And so Adam can just tell me, hey, Allison, I put that data in their GTEx comparison project, and you can go, and on my dashboard, I have it here right at the top. One of the things I want to talk about, since we're talking about GTEx and integrating other data sets, is that, you know, a lot of this data is controlled access. And me as a technical bioinformatician has jumped through a whole bunch of hoops to try and make sure that I have all the right dbGaP access and et cetera. And we really started thinking about that in the settings here where we have the different integrations. So we have both Kids First integration through partnership with the University of Chicago that allows us to handle the Kids First data, and really through a really strong partnership with the NCI Cancer Research Data Commons as well, having authorized studies here that initially we've been piloting around Target with the pediatric brain data, but this allows us to really have this idea of interoperable and collaborative data sets in line with Kids First, as Adam was saying, that, you know, there's probably other kinds of low-grade gliomas or other brain tumors that you might want to pull in here. So that's obviously a really important part of how we make sure this data is secure and maintained as we do these different handoffs between these teams. So if I go back to my dashboard, and I have many Chavatica projects, as you might imagine, so sometimes it takes a little bit longer to load. But I can just click on it directly, and it takes me to the Chavatica platform. And as I was saying, again, Chavatica is talking to a trusted backend. You have your ERA Commons. This is talking to dbGaP, understanding exactly what data I have access to. So when Adam does that handoff and I log into this platform, it can check my different access. And here you can see in our pre-baked GTEx comparison, that, you know, me, Adam, and PJ Raman, who's one of the bioinformaticians who really does a lot of this work, set up in this project. And you can see here in the files, that the different files from Kids First that Adam just pulled over, the BAM and CRAM files are here in Chavatica. And this is a part we haven't yet pre-baked, but really once it's there, you might want to reanalyze it. So GTEx itself has different processing, has done different harmonization, but maybe I've already processed it with the Kids First pipelines, and I can start right away. But this is really what Chavatica allows you to do, is the kind of very technical bioinformatics work of finding the different specific workflows, running them at scale. We use the platform to run the DRC pipelines. We are able to do thousands of whole genomes a week on this platform. So we've been working to put the Kids First pipelines on here. So if you wanted to bring your own data, or you're pushing other data that was from NCI or other places, you could actually bring it in here, and with a few clicks, get set up right away to run that pipeline on your data, including all the different reference files. So these are the exact same reference files that we use to harmonize the Kids First data as well. And so this is really starting getting to that, you know, technical bioinformatics role of choosing all the different files, all the different parameters, and really working in collaboration to be able to take that handoff from someone who's looking at it from a more biology standpoint. And once you imagine I do all those workflows, I get all the real data, really then it starts being, okay, how do I collaborate back? How do I do the different visualization? This is where the data cruncher features come into play. And so now there's both Jupyter Notebooks as well as RStudio on here. And so you can see PJ, who's been doing a lot of this analysis, has set up a notebook that is really starting to compare different BRAF fusions with pediatric low-grade gliomas. And you can have a bioinformatician come in here, write all the different R, and then have the nice visualization and plots that I can then hand back over to Adam, who can then look at the different biological insights there.
You know, we had some analysis that already were done and are available for exploration, for example, in a platform like the cBioPortal platform. But obviously, you know, when you test hypothesis, you end up having to do some analysis. And as Alison highlighted, oftentimes when you start integrating data sets, either public data sets like GTEx with your own analysis, you have to harmonize such data in order to try to drive the interpretive space. You know, based on just that initial variable representation of fusion genes in low-grade gliomas and non-low-grade gliomas, I asked PJ just for this demonstration to just run a comparison of low-grade gliomas versus non-low-grade gliomas, and then compare those expression profiles against GTEx. But the main purpose here to say, you know, the samples that have birth defects don't have a canonical target like a BRAF fusions, can we maybe identify other overexpressed genes in that target? And so here, as I'm scrolling down, is the code. And you can see even here that if you compare, and here this is just the log p-value of the expression, BRAF fusion negative versus positive low-grade glioma samples, you see, you know, that there are many more overexpressed genes in the context of the BRAF fusion positive samples, and that the BRAF fusion negative samples, of which the birth defect affected sample are clearly different than the BRAF fusion positive one. And once you have that, you can actually take those data, compare them to the harmonized data in GTEx, and then begin identifying genes that may be differentially overexpressed or underexpressed in different tissues versus the tumor sample in order to really assess you know, what new dark target you might want to pursue. Here we just chosen as example MDK, which you can see has low expression in some tissues but higher in some others. So if you were to design a target, you'd be aware of the potential toxicities associated with it. So hopefully this provides you, you know, the right vignette of the kinds of workflows that you can do as a team, leveraging your differential expertise from both your domain-specific or disease-specific expertise, plus technical, informatic, and platform-based resources.